Hey friends, welcome to this channel. If you're new here, I am Indrani from Bong Biology, and this is the endoplasmic reticulum part 2 video where I am talking about the protein sorting via endoplasmic reticulum. So in the previous video I have talked about the signal sequence and all. So signal sequence is a stretch of amino acid, around 20 to 25 amino acid, and among them 7 amino acid are totally hydrophobic. They are always present at the N terminus end of the protein, and they are the main characteristic features of all the secretory proteins. Now we'll see how these secretory proteins get translocated within the endoplasmic reticulum lumen. So before starting, I have to consider some facts that signal sequence is recognized by SRP. This SRP is signal recognition particle, and this signal recognition particle, they contain 6 polypeptide chain and a small RNA that is called SRP RNA. Okay, so let's say this is the signal recognition particle, and on the endoplasmic reticulum membrane we have another thing that is called SRP receptor. So this signal recognition particle will go and bound with this SRP receptor, but before that this signal sequence actually identify this signal recognition particle or SRP actually identify the signal sequence and bind to this signal sequence. So when signal sequence is recognized by this SRP, this whole compound will go and attach to the SRP receptor. So this is a fact, just remember this fact. Okay, now let's go back to our part. So now this is, here we are talking about co-translational translocation. That means the translocation is going on when the peptide synthesis is not completed. So what happened is, first the signal sequence is emerged from the ribosome, and as the signal sequence emerges from the ribosome, it is recognized and bind by the signal recognition particle. This is the signal recognition particle. Now this SRP escorts the complex to the endoplasmic reticulum membrane, where it binds with the SRP receptor. This blue thing is the SRP receptor. This SRP is then released, and the ribosome then binds to the translocon. This thing is the translocon. This is the translocon by which the translocation will be done, and the signal sequence is then inserted within this translocon. This is the signal sequence, and this is now inserted. One thing I forgot to mention, that when SRP bind with the signal sequence, it immediately inhibit the translation. So the translation or the peptide synthesis is stopped by SRP when SRP bind with the signal sequence. But when SRP is released and the ribosome is now bound totally with the translocon, and the signal peptide enter within the translocon, the translation can resume. So the translation resumes, and the growing polypeptide chain is translocated across the membrane. This growing polypeptide chain is translocated across the membrane, and finally there is a cleavage of signal sequence by the signal peptidase. This is the signal peptidase, and thus the polypeptide is released into the endoplasmic reticulum lumen. But what happened, this is co-translational translocation, right? But what happened in case of post-translational translocation? This is post-translational translocation. That means before translocation the translation of the peptide chain is totally completed. Now, the proteins destined for the post-translational import to the endoplasmic reticulum are synthesized on the free ribosomes. These are cytosolic ribosomes, and these proteins are maintained in an unfolded state by chaperone. These chaperones are HSP70, that means heat shock proteins. And these signals, they also have their own signal sequence, and their signal sequence are recognized by Sec62-63 complex, which is associated with the translocon of the endoplasmic reticulum membrane. Okay, this is the translocon and this is Sec62-63 complex. The Sec63 protein is also associated with a chaperone which is called BIP. This BIP is again HSP70, and this BIP acts as a molecular ratchet to drive the protein translocation into the endoplasmic reticulum. So this is the difference between post-translational translocation and co-translational translocation. Hope you understand. Next, till now we are discussing about those proteins which are destined into the lumen of the endoplasmic reticulum, and so they will be destined further to the inner part or the luminal part of Golgi apparatus and lysosome and endosome and so on. But what happens if a protein should be associated with the membrane? So what happened in case of membrane proteins? So for membrane proteins there are three types of orientation, right? And the membrane protein should always contain the alpha helical part. The alpha helical part is obviously at the transmembrane part is the alpha helical part, and in this alpha helical part it should contain at least 20 to 25 hydrophobic amino acid. So it should be hydrophobic amino acid for this transmembrane part, and there should be two kinds of orientation. One where N terminus is associated at the cytosolic part and C terminus is associated in the luminal part. The second one is just opposite, where C terminus is in cytosolic part and N terminus is in lumen part. For both these cases we can see that they have one transmembrane part, one transmembrane domain. Hope you can see this red color. So they have one transmembrane domain, but in this case we have multiple transmembrane part or multiple transmembrane domain. So how this things happen, or how these proteins are translocated into the endoplasmic reticulum membrane, we'll discuss it about all this. There are two kinds of signal. One is stop transfer signal and another one is internal signal sequence. We'll discuss it but in the next video. Okay, so hope you enjoy and hope you can understand everything over here. If there is any question, please let me know. Okay, so bye.